Hey, let's learn about protein synthesis today. I know you might recognize my voice, and I bet this isn't the first time I'm going to teach you science, so let's get started. Here is a cell. It is composed of a cell membrane made out of phospholipids and proteins. The side of the cell is filled with a gelatinous liquid called the cytoplasm. Within the cytoplasm, the cell houses imperative organelles like the nucleus, the endoplasmic reticulum, the Golgi bodies, and many more depending on the type of the cell. Protein synthesis begins with the DNA that is located inside the nucleus and chromosomes. The DNA has two ends, a 3' end and a 5' end. The DNA is unzipped and the hydrogen bonds are broken with an enzyme called helicase. This creates two strands of DNA. The bottom one is called the template strand, and the top one is called the coding strand. This region of the DNA, where the mRNA will be made, is known as the operon. Then another enzyme called RNA polymerase creates an mRNA copy of the coding strand. This enzyme attaches right after a section of the DNA known as the tau box. This entire mRNA strand is an exon, which is the part that leaves the nucleus. But before leaving the nucleus, it needs two other things attached to it, a 5' cap and a poly-A tail. Alright, now the protein travels through the nuclear pore to a ribosome located in the magnificent endoplasmic reticulum. Ribosomes are protein-building factories that use tRNA molecules to actually create the polypeptide chain of amino acids. This is a process known as translation. Ribosomes are made of a large subunit and a small subunit, where the tRNA molecules bring in amino acids to eventually make a polypeptide chain. The amino acid is attached to the tRNA at the tRNA amino acid complex through a protein called aminosyl tRNA synthetase. These tRNA molecules go through three different sites on the ribosome. These are the A site, B site, and E site. The tRNA attaches to the mRNA using its anticodon, which attaches to the codon on the mRNA. Translation begins with initiation, which is when the tRNA brings in the first amino acid, methionine, which attaches to the start codon, which is AG. Then this tRNA moves along to the P site. At the same time, the next codon is being read and a new tRNA is brought in as well. The amino acids join at the P site, where they form peptide bonds with each other through a protein called peptidyltransferase. This process of making the polypeptide chain longer with additional amino acids is known as elongation. After the P site, the tRNA moves along to the E site, where it finally unbinds from the mRNA and flows off into the cytoplasm. This process is repeated for all the codons on the mRNA until the stop codon is read. No tRNA anticodon matches up to the stop codon on the mRNA, which signals the ribosome to stop translating. This process is known as termination. After the polypeptide chain has been made, it goes through structure changes. The formation of alpha helices and beta sheets is called secondary structure. The polypeptide chain also forms disulfide bonds, which are formed between two cysteines. These bonds cause tertiary structure folding of the polypeptide chain. After this, a polypeptide chain heads off to the Golgi bodies to go through some post-translational modifications. Some post-translational modifications include glycosylation and ubiquitination of the protein. Firstly, during glycosylation, various carbohydrates can join to the protein and create glycoproteins. Repulsive carbohydrates that might participate in glycosylation are galactose, glucose, and fructose. Next, the polypeptide chain can also go through ubiquitination. In this process, small proteins are attached to the protein that might serve imperative functions such as ATP regulation. The finished protein is put inside a vesicle that carries it through the cytoplasm to the edge of the cell membrane. Finally, the protein diffuses out of the cell membrane, which is known as excitosis. This protein will now go perform some important function to keep you and all the diverse life around us living.